Hello, welcome to some video notes on topic 2. Today we're doing 2.4. When we're doing the first part of 2.4, we'll be getting into the structure of proteins. And so we will talk about a little bit more information about the monomers that proteins are made out of, which are amino acids, and how we get so much variation of our proteins from those amino acids. A little bit about some rules about how proteins are created that all living things seem to be following. And then finally, we'll get into the very, very detailed, important slide where we talk about all of the structural levels of a protein and what all those vocabulary words are that you need to know.
No, God! No, God, please, no! 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 No!
So this first section is mostly going to be following the first parts, the first learning objectives that we have here. We're going to be talking about the amino acids. We'll be talking about the way they're linked together. We're going to talk about different size of proteins, the different properties of amino acids, talk about how amino acids are put together to make proteins using DNA as the genetic kind of blueprint. But most importantly, we'll be talking about the structure of a protein in order to create its three-dimensional shape and ultimately its function. So let's go back and review some concepts if you don't quite remember them. So if we're talking about the building of proteins, we'll be looking at something that is anabolism or anabolic reaction through condensation reaction. So if you remember, condensation reaction means water is being formed. This water, of course, is coming from the hydrogen of the amino group of one amino acid, the second amino acid, and the hydroxyl group here on the carboxylic acid side of the first amino acid. And so when this occurs, we get water and what we call a peptide bond. And so proteins can be also referred to as polypeptides because they are made of many peptide bonds. We could also, if it's just two amino acids, you could call it dipeptide. If it's three amino acids, you could call it tripeptide. But really, once we get beyond simple combinations of amino acids, we just start calling them polypeptides because there's just so many of them. And of course, this entire reaction is going to occur inside of the ribosome. The ribosome's function inside of a cell, if you remember back from topic 1, is to create proteins. And so it will use genetic information in the form of mRNA, which we'll talk more about in topic 2, as a guide in order to assemble a polypeptide by putting the correct amino acids in the correct order and then fusing them all together in order to make a long chain of amino acids that eventually folds into a three-dimensional protein, which we'll get to about the three-dimensional structure a little bit later in this PPT. So these bonds are all formed, and remember they're all covalent linked bonds, and remember the idea of monomers versus polymers. Monomers are our single units, so a protein is made out of monomer units, which are called amino acids, and therefore a polypeptide would be considered a polymer. So if we were to look at a ribosome going through its function, here's a three-dimensional structure of a ribosome, what it looks like inside of the cell. And so what actually is going to happen is our ribosome is going to be attached to something called messenger RNA, or mRNA, which is made up of nucleotides, or A, T, C's, and G's, right? The things that DNA and RNA are going to be built from, or A, U, C's, and G's, because it's RNA. And it's going to read it in little 3 base pair sections called codons. And so that 3 gave us this amino acid here, and this 3 is giving us this amino acid, and this 3 is going to lead to this amino acid, and then this next 3 is going to be leading to this next amino acid. And so we're going to talk in more detail about this overall process in another video, but essentially using the code and matching or base pairing so that we kind of read the code or decode it using kind of a rule with base pairing rules. The correct amino acids are put in order inside of the ribosome, and then one at a time, a peptide bond is going to be formed between those amino acids. And so we get a nice long chain of a polypeptide as it grows out that direction. And as we see, eventually, once this is finished, the ribosome will release the protein, and then it will not sure if the slide just cut off that sound. I know what I was saying is that as the chain gets released from the ribosome, it will eventually fold to create our functional protein, which we'll talk about a little bit later in this video. Now, if you remember, there are 20 naturally occurring monomer units that make up a protein, and those are our 20 amino acids, basically. And so you don't have to know all the amino acids here. You don't have to know the structures. That's don't worry about that. But you should know that some of their properties, or some general properties, exist. So we have ones that are going to be positively charged and ones that can be negatively charged. And so being charged, we could also refer to them as being polar amino acids. So they would be more likely to interact with water, right? Because they can dissolve into water being polar. We also have polar uncharged ones, so they're polar, but they don't necessarily create a positive or negative charge, so they still can mix with water fairly well. So these would all be like hydrophilic ones. We can also have some sulfuric containing ones, or ones that contain nitrogen chains. These can be polar as well, or sometimes nonpolar, but they can produce very special bonds because they contain sulfur, and we'll talk about those bonds a little bit later in the PPT. And then there's also a large group that is going to be nonpolar. And so if you remember, nonpolar meaning that they will not mix with water, they will be hydrophobic. And so the nonpolar ones would be more likely to be found in the middle or the center part of a protein, because it's trying to avoid being around the water as much as possible. And it would also be located in a membrane. If you remember that a cell membrane has a hydrophobic section in the middle where the phospholipid tails are located. So since that is a hydrophobic section, the polar amino acids really wouldn't be able to fit in there very well. They wouldn't make the right shape protein. But the nonpolar ones would do just fine. And so, for example, our transport proteins that we see, like our ATP, sodium potassium pump for active transport, or just a sodium channel or a chlorine channel that will move through a protein or through the membrane, those proteins would be having a bunch of nonpolar amino acids in the middle section that's actually inside of the membrane. So we're going to get more into this a little bit later when we talk about the physical structures of how proteins are made. So even though we do say that there are 20 naturally occurring amino acids, there actually is a 21st amino acid, a very special one called hydroxyproline. And hydroxyproline is basically a modified proline. And so what happens is that a special enzyme called prolyl hydroxylase, right? And so it can give an idea that it ends in ase, right? So we know that it's an enzyme. Hydroxyl and prolyl, this idea that it's creating or adding a hydroxyl group to a proline, right, is where this name is basically telling us what it does. And that is what it does. So it will take a proline and it will convert it into hydroxyproline through this reaction. And so we can just simply modify the protein because it increases stability when we're talking about fibrous proteins like things like collagen, and it can help increase tensile strength. So your tendons, your ligaments, your skin, the walls of your blood vessels, these are all things that are supported with collagen, which is a special elastic protein. So your parts of your body are very capable of stretching fairly large distances, but then their ability to be elastic and then return back to that shape, or mostly back to that shape, is an important feature. And a lot of this comes from this hydroxyproline. So there are 20 naturally occurring amino acids, but there can also be one other amino acid that can be modified in order to improve its function. And there, who knows, maybe there will be other amino acids that we discover in the future. Maybe that's not just these 20 or 21, depending on how you want to think about it, that exists. However, currently, from what we can tell, the naturally occurring ones that you will find being used in bacteria and fungi and plants and animals and all forms of life, these are the 20 ones, or 20 of them. You do not need to know their names, so don't worry about that, and you don't need to know their structures. You do need to know the general structure of amino acid, right, when we have that R group, which is where we get the 20 different forms, is by changing that R group. And you need to know their general properties, like things that are polar versus nonpolar, things that contain sulfur versus ones that might be positive or negatively charged. So hopefully that makes you relax a little bit about what information you actually need to be able to produce in the future. Now, the thing is that a lot of proteins, there's a lot of different proteins that exist inside of your cells and keep you as a functioning organism. Just to kind of give you an idea, I just talked about collagen, right, which is one example of an elastic protein that is used in lots of different parts of your body. But one of the most common forms that proteins end up taking would be that of enzymes, right? And enzymes are those biological catalysts. They help power those chemical reactions that are necessary to keep us alive. Now, there are about maybe around 10,000 different types of enzymes that can exist inside your average animal cell. So many different enzymes because they're all there to be in charge of very specific chemical reactions. They kind of do maybe one or two really specific jobs, and that's it. So in order to have all these very different chemical reactions all happening at the same time in different places all over the cell, you need lots of different types of enzymes to drive those chemical reactions. So that's just the enzymes are in the thousands to tens of thousands in terms of numbers. If we were to talk about the number of different proteins in total that could exist inside of a single animal cell, it could be in the hundreds of thousands. So if we're only using these 20 amino acids, how are we getting hundreds of thousands of different proteins, different types of polypeptides? Well, that comes from the properties of the amino acids, right? The number of the amino acids, and then, of course, the different types or the sequence of those types of amino acids. So, for example, here we have a bunch of amino acids, and we're showing them as being different amino acids by having different colors, right? So the fact is that amino acids, or sorry, polypeptides can be any number of length built from any combination of these amino acids and in any order. So let's just look at these two chains right here, right? So from these two chains, we see that there are here, and this one, can't see very well, there are 7 amino acids. And here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Here we have 10. I counted that out right. That's 7 amino acids and 10 amino acids, right? And you can see that the number of the different amino acids that occur inside of them are different. Here we have 2 of these peach-colored ones, right? Here there are 2 purple ones, versus the other one there are no purple or peach ones. Here there are 3 kind of grayish ones in a row, and here there's only 1, right? So there are different numbers of the different types of amino acids. There's a different number of amino acids in general, 7 versus 10, and the sequence is different, right? We're starting with blue, yellow, purple, gray, where the other one starts with red. Actually, looks kind of like a white. Red, white, gray, gray, right? So there's a lot of differences between them. So think about it. Let's just ignore the polypeptide of 10. Let's just focus on the shorter one, which has 7. If we took those 7 amino acids, because there are 20 different amino acids in existence, that means we have 20 to the 7, the power of 7, possible combinations that could exist. We could have 7 blues. We could have 7 yellows. We could have 7 purples. We could have an alternating blue, yellow, blue, yellow, blue, yellow, or an alternating yellow, purple, yellow, purple, yellow, purple, right? There are so many different combinations of just these 7 amino acids. So we get about 1.2 billion combinations from this, right? So that's hopefully maybe giving you an idea of why we can have so many different types of so many different types of polypeptides, because we have a lot of monomers, like 20 different monomers is a good number of different monomers, but it's also the length of the polypeptide and the specific sequence of the polypeptide, which eventually causes it to have specific properties. And so that's where all the variation comes from. So right now, maybe a polypeptide has about 30,000 amino acids in a chain. So that means of that 30,000 amino acids in a single polypeptide that we're talking about, a complex polypeptide, there could be an almost infinite number, all right, of combinations. There's so many combinations, and it would take more time than the existence that we have in nature to create every single one of those combinations, even, you know, inside of a cell. It's just an infinite number, really, in some ways. So all this variation is where we get all the different types of polypeptides, and where we all get and all those different proteins are able to, you know, keep us alive and do all these different specific functions. So earlier we talked about the fact that a ribosome is going to be responsible, right, for making a polypeptide. They're going to do what we call polypeptide synthesis or protein synthesis. And they're using messenger RNA or mRNA as a template. They're using it as a guide to know what should be the order of the specific amino acids, how many of which amino acids and which in order does it need in order to make a very specific protein. And cells need to make very specific proteins over and over and over again, and they need to be very exact, right? They don't want to make many copies of the same protein and have them not all work properly because there was some issue with the production of the proteins somewhere in the process of the ribosome doing its job. So the ribosome is very, very thorough, trying to make sure that it follows the mRNA to make the exact protein that is necessary. So then, where does this mRNA come from? So now we're going to just talk a little bit about what we call the central dogma of genetics, or really the central dogma of biology. And that's based around these core principles. First off, information in living things is stored in the form of DNA. That is the function of DNA. It's the blueprint or the guide for making a living thing. So in DNA, we would say that there are genes. There are specific sections of the DNA that produce a very specific trait or a very specific protein. Sorry. DNA is stored inside the nucleus in our eukaryotic cells, so it's a little bit protected. And so for DNA, it's going to have to be removed from the nucleus, but we don't take the DNA out, but we want to take the information out. So rather than taking DNA out of the nucleus, where it's more likely that something could happen to it, we copy the information from DNA into a form that can easily be used and then, degraded and recycled, and it doesn't really matter what happens to it. And so this copied form of the information is called RNA. Specifically, we call it messenger RNA. It's taking the message or the information from DNA and taking it out of the nucleus so that the ribosomes in the cytoplasm or the ribosomes in the rough ER can do their job, can make a protein. So this entire process is called transcription, and we're going to spend a whole lesson just talking about transcription and how transcription occurs and what is happening during this process. Once the mRNA gets out into the cytoplasm, it then is going to interact with a ribosome so that it can be converted into a polypeptide. So this process will create what we call the product or the protein. And this process is called translation. And translation is when mRNA is then used by the ribosome in order to create a polypeptide. And again, we will have a whole class talking about translation and how this whole process occurs. But the thing is that as far as we can tell, this is the rule that we see all living things follow. All living things store their information as DNA. They go through the process of transcription in order to change the information or copy the information in DNA into a new form called mRNA. Then mRNA is going to be translated by a ribosome into a polypeptide sequence. And then that polypeptide eventually creates the protein, which is, doing some type of specific function inside of the cell. So when we come back to this idea of transcription versus translation, right? Transcribe something is very much to copy it, right? DNA and RNA are both nucleic acids. They're both the same type of code. So the information in DNA is being copied into RNA. So that's why it's called a transcription, because it's a copy. The word translation, which you guys have probably heard before, this idea of going from one language to another language is to translate it, right? Even though they might be based around, similar ideas, English is not the same language as Spanish, and Spanish is not the same language as Chinese. So because there are different rules, there are different sounds, there's different grammar, we are translating it. We're taking the general information from one form and we're changing it into another form, but trying to keep that information about the same. So since RNA is a nucleic acid, but polypeptides are proteins, proteins and nucleic acids are not the same things. So we are translating the information in mRNA into a new form, which is the polypeptide sequence. All right, so that's why we call it translation. So we'll talk about both of these in more detail in another set of notes when we'll get into each translation and transcription about what's currently happening in them. So summarize again, genes are the codes for making polypeptides, information stored in the nucleus in the form of DNA. We need to get that information to the ribosomes in the cytoplasm so they can be created into polypeptides. Messenger RNA is our go-between, and so everything follows this rule of genetic information in DNA being converted to mRNA, and then the mRNA being translated into a protein or an amino acid sequence in order to create a polypeptide. So again, just to emphasize this, pretty much all organisms are following this genetic code, this rule. The only things that really kind of go against it would be viruses, but viruses, there is a debate whether or not we even could consider them to be living things. So pretty much all organisms are using DNA in the same way, are using mRNA in the same way, and are using amino acids in the same way. So this is kind of a universal thing. But we get all this variation in our amino acids. We get all this variation in, in our proteins from all the different amino acids being used in different numbers and different sequence. And that, of course, is the reason why we have so much variation in living things, is ultimately because of this variation in our proteins. Okay, so we are almost done. Now we're going to talk about how we make a polypeptide and then how a polypeptide eventually gets folded into a functioning protein. So again, just to remind you, polypeptides is this idea that we've taken many peptides, or many amino acids, and we've put them together creating peptide bonds. That's where we get polypeptide, or many peptides. So this is all done through a condensation reaction, meaning that we will take the hydroxyl group from the first amino acid, the hydrogen from the second amino acid. They get removed to create water, and in that bond between the two amino acids, we get our peptide bond, right? So there being 20 different amino acids, the combination of the order that they're put in, the exact sequence, the exact numbers, the fact that some of them are polar, some of them are non-polar, some of them are positive charged, some of them negative charged, some of them contain sulfur. All of this leads to the variety of how a protein will be formed. So a polypeptide is technically just a chain of amino acids all connected to each other. A protein and a polypeptide, even though we might use them interchangeably, are not exactly the same thing. A protein is when a polypeptide has properly folded into the correct three-dimensional structure in order to have it be a functioning material, some type of functioning enzyme or some type of structural protein, doing whatever its job it's supposed to do. Until the polypeptide has properly folded into the right shape, it's not actually a protein. So what we're going to go through is how do we get the polypeptides to fold into a protein? What are the levels of complexity when looking at the transition from polypeptide into a protein? Okay, so you might have to go through this last part several times. You might need to pull this up, on another screen somewhere to look at the. Okay, so there's a lot of information right at the end, but hopefully you can go back and watch it again if you're confused. And if you have any questions, you can let me know.